All research in my laboratory focuses on one major question. How does a neuron choose the correct synaptic partner to ensure appropriate function for a neural circuit? One long-standing hypothesis states that synaptic partner matching occurs through cues present at the growth cones of developing axons and dendrites. My lab seeks to identify these cues and then modify them to enable engineering of neural circuits in vivo. We conduct this work in the fruit fly, Drosophila melanogaster. During development, synapse formation in the fruit fly utilizes mechanisms conserved in mammalian systems. Additionally, a genetic engineering toolkit and relatively small number of synaptic connections make the fly ideal for understanding how synaptic partners are established. Our work will provide an enriched understanding of how neural circuits are organized to drive behavior and how faulty partner cues contribute to neurodevelopmental disorders.